like to thank the organizers for invitation, and it's a pleasure for me to be here and take a few words about my experiments. I work in the Institute of Immunology and Experimental Therapy in Polish Academy of Science, and I would like to talk about the role of microRNA 125p in calcitriol-induced differentiation of human leukemia and lymphoma cells. In the last year, leukemia and lymphoma caused about 7% of all malignant in adults. These diseases are more often in men than women and usually arise after the age of 60. And during 4 years, overall leukemia incidence rates increased by 0.5% per year. Cancers which occupy the first place in children and adolescents are acute lymphocytic leukemia and Hodgkin lymphoma. But there are also other types of leukemia and lymphoma which cause about 40% of all cancer incidence in children and adolescents. Leukemia is a cancer of the bone marrow and blood, and all formed blood elements arise from one bone marrow stem cells. These stem cells divide into two types of specialized stem cells, myeloid stem cells and lymphoid stem cells, which differentiate into mature and functional cells. So characteristic abnormality in leukemia is blockade at the early stage of their development and failure of differentiation into mature cells. So hematological malignancies are classified into 4 main groups according to the cell type and rate of growth. And there we have acute lymphocytic, chronic lymphocytic, acute myeloid, and chronic myeloid. But these 4 basic groups are divided into many subgroups, but all the time the new information are accumulating and this classification is changed. But the VGO classification system of leukemia and lymphoma are based on the published data for better recognition and treatment of these diseases. And the main factors which causes leukemia and lymphoma are genetic aberration, which leads to changes in signal transduction, transcription, and growth factor expression changes. And nowadays, much attention has been focused on the epigenetic disorders, which include microRNA. And we know that microRNA are responsible for cancer development, and also they have important role in the development of chemosensitivity and chemoresistance in cancer, including leukemia and lymphoma. And they repress very many important cancer-related genes, what prove usefulness of these molecules in the diagnosis and treatment of cancer. And the main goal of treatment of leukemia and lymphoma is to destroy the leukemia cells and allow the normal cells to form in the bone marrow. And standard treatments include the standard protocols of chemotherapy, and usually there are continued up to 3 years. But when there are no signs of leukemia for 5 years, a person is usually cured. But if the leukemia doesn't go into remission, or if comes back within the first few years, treatments may include more chemotherapy, a stem cell transplant, or joining a clinical trial for new treatments. And one of compounds which has been mainly successful in hematological malignancies clinical trials is active form of vitamin D calcitriol and its analogs. And the role of the vitamin D in the inhibition of malignant cell proliferation is indicative of its future use in cancer therapy. And everything was started 30 years ago when attempts have been made to treat very different cancer types with differentiation-inducing drugs. And to date, the only one successful drug is used in differentiation therapy. It calls all-retinoic acid, but it results often in differentiation syndrome named retinoic acid syndrome. But searching on differentiation-inducing drugs leads to discover the active form of vitamin D calcitriol, which induce differentiation of murine leukemia cells, and these murine leukemia cells differentiate in vitro into major macrophages. But there is the issue of hypercalcemia, which occurs when calcitriol concentrations range increase, so it limits the calcitriol clinical application. So because of that, the developing of analogs was started, and one of active and very good analog is calcitriol, which is used in treatment of hyperproliferation diseases like psoriasis, and it has reducing mobilizing properties of calcium. Calcitriol activity occurs via two pathways. One is slow genomic response, and the second is rapid non-genomic response. And in these two pathways, calcitriol binds to the vitamin D receptors, and in the genomic pathway, this ligand bound binds to the partner receptor, retinoic X receptor, which influence on the vitamin D receptor elements, which are localized, which binds with high affinity to the promoter region of genes, which are crucial in many actions in cells. And the second way of calcitriol action is rapid non-genomic response, where calcitriol binds to vitamin D receptor and activates many cascades like protein kinases, mitogen activated kinases, and phospholipases. But the main properties of vitamin D action is recurrence of co-activators, of co-repressors, which positively or negatively regulates the gene expression, which are involved in proliferation, apoptosis, differentiation, inflammation, invasion, metastasis, and angiogenesis. And the most action of calcitriol is mediated by the vitamin D receptor. But inside the cell, there is also another enzyme which could bind the calcitriol, and it's called CYP24, and its role is to make the calcitriol from active form to inactive form. So it reduces the calcitriol activity in cell. And this enzyme is a mitochondrial enzyme which is expressed in cells which are responsible for calcitriol and its analogs. But the main point is that both VDR and CYP24 protein could be regulated, and there are many suggestions that they are regulated by microRNAs. MicroRNAs, there are structures which in proper translation, processing, and blockade of mRNA type genes results in normal rates of cell growth, proliferation, differentiation, and cell death. When the microRNA are blocked, these microRNAs which function as tumor suppressors inside the cells, when they are blocked, it results in tumor formation. But on the other side, when the microRNAs are are amplification and overexpression, it could be also results in tumor formation. And one of the most interesting for our research microRNAs is microRNA 125. This is a family of microRNAs which are involved in many biological processes like proliferation, metastasis, immune response, and differentiation. And was the most interesting for us was a study created by Mori, in which he observed that microRNA 125b reduced endogenous VDR protein level, and also the effect of calcitriol was abolished by overexpression of this molecule. So we decided to check if in human leukemia and lymphoma cells, if these two molecules, CYP24 and VDR receptor, are expressed, and if there is any correlation with microRNA 125b expression. So firstly, we treatment these cells with calcitriol and its analog, and we observed that only 3 from 9 tested cell lines were sensitive for this calcitriol. And the most sensitive cell was MV411 cells, and in the case of the other lymphoma and leukemia cells, we can measure only proliferation inhibition in the highest used concentration. It was 1,000 nanomoles. And we observed that in the case of the sensitive cells to calcitriol and calcitriol, the morphology of these cells was changed compared to the control. And what was the most interesting, that we observed that when the microRNA 125b expression level was high, the expression of CYP24 was downregulated. And the opposite situation was when the microRNA level was low, the CYP24 expression was upregulated. We didn't observe any correlation and any changes in this microRNA expression with VDR on mRNA and protein level. In the second sensitive cell line, LH16, we observed that treatment with calcitriol and calcitriol induced CYP24 expression after 24 hours of treatment. And the similar situation was with microRNA 125b. And in both cases, the level of these proteins rapidly reduced to the basal level, and no changes were observed after the first. And also the morphology of these cells was changed into the more mature cells. And in the last but not least, sensitive to calcitriol and calcitriol KHP1 cells, we observed that when the CYP24 expression was upregulated, the microRNA was downregulated, and it was confirmed after the 96 hours when the microRNA level was upregulated and the CYP24 was downregulated. And we also didn't see any correlation with expression of VDR and mRNA and protein level. In the case of the whole of lymphoma unsensitive cells, we didn't observe the expression of CYP24. Also, the morphology of these cells was unchanged, and we didn't observe any relation between VDR mRNA and protein level. it was stable. And the same situation was in the second unsensitive cells of Jurkat, where the morphology was unchanged and we didn't observe CYP expression. And also in the most unsensitive cells, U2934, where the CYP24 expression was undetectable and the morphology of these cells was unchanged. And to confirm these results, we are going to inhibit these molecules to confirm this observation. And to conclude, the most sensitive to calcitriol and its analog human leukemia and lymphoma cells express CYP24, VDR, and microRNA 125B, but in the most unsensitive cells like Dowdy, Jurkat, on U2932, we didn't observe CYP24 expression before and after calcitriol and its analog treatment. And the highest changes in the expression of CYP24 we observed in the most sensitive cell line NF411, where the fold change was about 25,000. And in leukemia and in human leukemia and lymphoma cell, however, expression was similar both in mRNA and protein level, independently of treatment. So this is our laboratory team of experimental anti-cancer therapy. We also collaborate with many research institutes like Pharmaceutical Research Institute in Warsaw, where Professor Andrzej Kutner synthesized calcitriol and its analogs. And thank you for your attention.
Any questions?